Good morning. Welcome, everybody. Welcome to our webinar. So this is the fifth in a series of webinars that we are organizing about high-performance computing, supercomputing, or HPC, as you want to call it. By we, I mean Lux Innovation in partnership with Lux Provide and the University of Luxembourg. Allow me to introduce myself for those of you that don't know me yet. I am Ramona Kaula, and I'm working at Lux Innovation and the Luxembourg Innovation Agency. And today I'm not here on the stage by myself, but we have a speaker. Eleftheria, would you like to introduce yourself briefly, please?
Thank you, Ramona. Hello, everyone. My name is Eleftheria Charalambous, and I'm doing a PhD on the microbiome, actually.
Thank you. So Eleftheria will present today her work in the field of health and how she used HPC and what were the advantages for her of using HPC and how she thinks it was a boost for her research. So let me introduce first to you the platform where we are doing the webinar today. So we would like this webinar to be as much interactive as possible. So don't hesitate to share your contact details, to present yourself in the chat box. You can use this space also for technical questions in case you have some problems with the platform, you don't hear us or you don't see the slides. Don't hesitate to post there. Then on the right side of your screen, there is also the questions tab for the questions on the content. So there you can type your questions about the content that Eleftheria will present or I will be presenting or anything that you'd like to see in the next webinars is also welcomed. If you would like to see any examples in specific sectors, we are happy to prepare it for the next webinars. Then we have the poll question, the poll section. We prepared some questions to understand who you are and what are your future programs in the HPC domain. So let's see these questions that I invite you to answer, to have fun with us, and also to understand, what are your interests. Let's see the first one. How did you hear about this webinar? So that we can make our reach out to our audience better. So through social media, through our emailing invitations, from Lux Innovation website or someone that you know at Lux Innovation, from our newsletter or from the press or any other sources. Then a second question, do you plan to use a supercomputer? And if yes, in how much time from now? Maybe you're already using one, but if you are not, you're planning to do so in 1 month, 6 months, 1 year, midterm, so 1 to 3 years, or long term, 5 years. Or maybe you don't know and you are here just to discover, explore this possibility. Then a third question, for what would you use HPC? For simulations and modeling, for virtual testing and prototyping, for big data analytics and visualization, for artificial intelligence, including machine learning, deep learning and other techniques, or for prediction and forecasting, or maybe for other. Don't hesitate to answer this poll. I will take some time at the end to present you the results, and so you have a general overview of what the other attendees are doing as well. So I will give you some time to answer these questions while I introduce you to our concept of HPC Thursdays webinar series. So we are halfway. As you remember, we've seen the benefits of using a high-performance computing supercomputer, the basic components of supercomputer. We had a session on simulations and one on artificial intelligence. Now we are having this one of today on health use cases. We have the next one on the 7th of April on finance, and we'll have one in April as well on digital history example and one example in public administration. And last session, the 8th one, to specific to discover Meluxina, this unique business-oriented supercomputer, and don't hesitate to register as you see the pop-up here. And save the date for the 12th of May. We will organize an event and a half-day event in the afternoon. So you will receive an invitation in a couple of days, but if you're here and you're interested in supercomputing world, just sign up on your agenda. The 12th of May afternoon, we'll have something special for you. So after this presentation, let's see what Eleftheria prepared for us today. I will leave the floor to her to present to you her experience with HPC. Eleftheria, the floor is yours. Thank you for accepting our invitation today. So I will stop sharing my screen and you can share yours. Perfect. We can see it.
Okay, great. So thank you, Ramona, for the invitation. I will start with a bit of my background and my profile. So who am I? My name is Eleftheria Charalambous. I'm originally coming from Cyprus. And 13 years ago, I moved to the United Kingdom to pursue my studies, where I did a bachelor's in biomedical sciences, a master's in immunology and immunotherapy. Then I did some work experience as a lab technician at the King's College Hospital in London. And 5 years ago, I left United Kingdom for lovely Luxembourg, where I came to work as a biorefinery assistant scientist at the EPBL. And then a bit later, January 2019, I actually moved into the Department of Infection and Immunity to start my PhD with my supervisor, Dr. Jonathan Turner, with a micro-DTU project which investigates the microbiome in early life adversity. Up to now, I suspect you must have noticed I had no computational background. The introduction to the computational side actually started about 10 months into my PhD. And then recently, I also paid a visit in Ireland, where I went as a part of my project, basically, to have a visiting PhD researcher position in order to do modeling in the post-early life adversity oral microbiome. This happened quite recently. I went there in October, thanks to a grant, external grant I won with the Pelican Foundation. So now, let me explain to you what I'm actually working on. My thesis investigates the microbiome within the early life adversity concept. And the overview of this consists of multifactorial adverse conditions that are known to shape the epigenome. And these factors, they occur very early in life, starting from conception to birth and then following up to infancy. All these adverse factors are able to influence the balance between health and disease in later adulthood. And also, they are known to shape the epigenome. So the big question I'm looking into is where and how the early life microbiome stands in this concept. Could these changes happening via the microbiome, what could be the potential link and role of the microbiome here? And now, what do I mean when I say early life microbiome and early life adversity concept? So we start from during the fetal development, right after conception. Everything that the mother is exposed to, the nutrition, stress, possibility of diseases, medication, and antibiotic usage can affect the course of pregnancy, but also the microbiota of the mother. And then we're coming to the point of birth, which the mode of birth is what it will define what composition, the early life microbiome composition the newborn baby will receive. Then this composition during the first 36 months of life of the baby will be further shaped and developed, and it will be impacted by the living conditions that the baby will grow up into, by exposure to infectious agents, polluted and toxic environment, and of course the nutritional intake that the baby will receive. In my project, I have been working on people who have been adopted into Luxembourg from third world orphanages. So these people in their early adulthood were recruited as part of a cohort into Luxembourg in order to study how the orphanhood they experienced in their early life and the living conditions that they grew up into at that stage shifts their health and disease balance. It's also worth to note that the actual early living environment that these adoptees were brought up to covers all the factors of the early life adversity concept and includes the prenatal exposure, the institutionalization, social stress, exposure to infectious agents, the toxic environment. In order to ensure that we have high quality data and robust results, I have set up a pipeline that covers all the aspects of my data, which includes from collection, generation, and analysis. And that we start with the source sample collection. My pipeline is adapted on saliva, buccal cells, and fecal pellets, from where we take the source sample and then we do the microbial DNA isolation. And then from the microbial DNA isolation, we prepare the libraries and we perform 16S sequencing. It's worth to note that these first steps that I just described, it's the part where it's mainly the wet lab of my project, of my pipeline. And then in order to take the raw data and analyze the raw data from the instrument, from the sequencer, it's actually where my journey with the high performance computing started. From here to start performing 16S sequencing analysis, and then further data integration, visualization, and processing. When I talk about further data integration, this is important because in order to tackle what happens into these adoptees after what they experience, we need to look from a holistic approach, from like how all the factors that they're working together. So for this, I am actually working with microbiome data, with immune markers, metabolomics, viral infections, DNA methylation data, stress data, and lifestyle covariates. And of course, I'm also working on side projects on top of this, where I also integrate on all of these behavioral data and brain imaging. However, the important question today is, what is a wet lab scientist doing with the high performance computing? What you see in the screen actually sums it up. In a wet lab setting, what do? We work with tubes a little bit of liquid, and pipettes. But how do we know what is in the liquid? We cannot see it with the naked eye. But what is in the liquid, you can actually see it on the right side of your screen. The interesting part is what actually happens in between. So starting from a few microliters of starting material, and let's say minimum a concentration of 100 picograms of DNA per microliter, we are able to generate up to 10 terabytes of data, which then they need to be further processed. The sequences we generate need to be aligned, classified, and cleaned, and that will basically give us a new data set of 3 to 8 gigabyte of size, what we call it as the raw data. These raw data, they are not very informative by themselves. In order to make sense of what the raw data have and say, we need to integrate with what available clinical information we have. So taking all this clinical information, we are then able to actually see what is happening in the tube in an output of 3 megabytes JPEG that represents the biological feature. So basically, what is happening in the tube. And in the starting tube, what you see is a source sample which hides a composition of a microbiome, which then you just see it in a picture. So all this, back top to bottom, it's a process that will take, let's say, 5 to 7 days of lab work, 1 to 3 days of analysis. So let's say 10 days total to just get one single plot, which in the world of academia and science, this single plot is basically a small part of a figure, which is a part of a paper, and it represents, let's say, 3 sentences in your manuscript. So it will be like a 3 sentences part of my thesis overall. So back to the starting question, what is a lab scientist trying to do with the high performance computing? Well, trying to make sense of what comes out of the lab. And this is how I started. So thanks to the opportunity I had, and I started with the HPC setting, I was successful to translate what the saliva samples from the cohort I mentioned earlier were actually hiding. And thanks to the efficient analysis on the high performance computing, I was able to demonstrate that between our adoptees that you see as ELA and non-adoptees that you see as controls, the overall salivary microbial composition appeared to be very similar, at least at the phylum level. And the main and most abundant phyla, they were common between the two groups. And these were the Actinobacteria, Proteobacteria, the Firmicutes, and the Fusobacteriota. Now, if we want to start seeing differences between our groups, we need to dive into a deeper taxonomic level and a more precise taxa, the genus level, for example, where we can start seeing that we have differences in the most abundant genera. Furthermore, with the use of the high-performance computing, in addition of calculating the relative abundances, which tell you what is your composition of the bacteria that you have in your sample, we are also able to calculate what they are called compositional indexes. Such a compositional index is the alpha diversity. Alpha diversity is defined as the mean diversity of species in different sites or habitats within a local scale. In other words, imagine that you have a bag of gummy bears, like you see on the image here. And then how many different colors of gummy bears you have in your bag, it will tell you what is the diversity of gummy bears in your bag. And then what is the number of different colors of gummy bears you have in your bag, and if this number is even or uneven for each color, it will tell you what is the evenness of gummy bears in your bag. Such calculations are based on very complex mathematical equations, like you see, and actually they will take very long to calculate. But with the use of high-performance computing, this in a few hours, you don't just have the calculation, but you always have the actual visualization. And thanks to the possibility that I was able to do this on the HPC, I was also able to compare the diversity and evenness of two different oral communities, the buccal and the salivary microbial communities, which despite their very close geolocation, they're both located within the oral cavity, within the mouth. They're actually two independent and distinct communities from each other. Basically, thanks to the efficient processing that the high-performance computing offers, only in the third year of my PhD, I already got the most important key finding of my thesis, which is that indeed I can see differences between our study groups, between our adoptees and non-adoptees. So with the data I generated on the relative abundances of different bacteria within the samples, when we added that in our statistical model, we revealed that the individuals that had been exposed to adversity had lower relative abundances of specific taxa, like I explained earlier with the different gummy bears. So all of the individuals that experience adversity earlier in their life, they have less gummy bears of each color compared to the controls, the people that did not experience the adversity. So thanks to the possibility of efficiently using these calculations on the HPC, I was able to prove that we have an association between the microbiome and the experience of adversity early in life. Nevertheless, concerning my project, yes, okay, it is fascinating that we are able to see the same differences 25 years after adversity in adoptees coming from diverse national and cultural origins, together with many interesting associations we see together with the lifestyle and environmental factors and physiological covariates. However, if we want to see deeper into what is the actual role of the oral microbiome here, we still need to investigate what is the interaction between the microbiome, the immune system, and the phenotype, that's the ELA phenotype, what we call it. So in the scientific world, when we lack experimental samples, it means that it's time to run some simulations. And thanks to the possibility in performing constraint-based modeling, it is possible to take the relative abundances that I have generated already and generate further personalized models for each member of the cohort, which taken together, then it will give us the predicted metabolic profiles of each participant. And with this, we will be able to answer what is the metabolic impact of the compositional differences that we have observed and already published. And this is what I am currently working on. Overall, why is my project important? Because what happens in the first 1,000 days of our life is the strongest predictor of the disease we get later in life. Very important is that the microbiome component appears during the first 2 hits. As I explained at the very beginning, it's defined at birth and it's shaped through exposure to the first environmental factors. This microbiome component potentially plays an important role in maintaining the balance between health and disease. Then the second big question is why using the high-performance computing has been very important for my project. the most important reason actually is the ability to run and analyze a large sample size. If I would try to analyze my data on a laptop setting, the laptop will take approximately, let's say, 25 samples maximum, which is not a very good number. It's okay, but not for a big cohort. My cohort had 115 participants, and actually, because of the possibility to run this analysis on the HPC setting, the maximum number of samples I was able to analyze at once, it was actually 400 within the LIH cluster. And the second most important thing is that the larger your sample size is, the longer it takes to analyze. And the HPC allows for a very fast processing, and it's very time efficient. This is important not just for me and my project, but actually it's important for a lot of other projects and cohorts that they're working into studying larger populations. Everyone that is seen within the field, medical field and medical research, we all know that the golden rule of statistics is that the larger sample size, the more closely we approximate the population. So the possibility that we can do this within the high performance computing setting, it's great. It's one of the greatest. So the HPC has actually become popular because of the COVID-19 pandemic. Its usage had a tremendous impact on the COVID-19 research and vaccine and drug development. Some examples of its usage include the whole Earth pandemic modeling, forecasting for human sensitivity to infection, looking at the impact of viral mutations on infectivity, and investigating the evolutionary path of viruses. And in addition to the massive impact on COVID-19, it is also very useful for the overall drug discovery, design, and development procedure. Further applications also include what is known as the virtual human, which technically allows the generation of 3D virtual clones of patients, allows dynamic simulations of muscles and joint interactions, and even bone implant modeling. In general, the HPC usage is paving the way towards the personalized medicine and digitalization of medicine. That was all for today. I hope more scientists will continue decoding genetic information by joining the coding world. And what I know for sure is that my journey with the HPC will definitely continue. Thank you very much. And thank you to Lux Innovation for this opportunity, and our audience for their time. I am happy to answer any questions that you might have.
Thank you, Eleftheria. That was an amazing presentation where you highlighted very well what was the advantage for you to use HPC and what allowed you to reach with the results that you obtained by using the HPC. So I invite our audience to post the questions for Eleftheria and for me when in the Q&A session that we'll have afterwards. And now I would like you to introduce the National Competence Center and the support services that we offer to help you approach the supercomputing world and to explore how you can apply supercomputing resources to your activities to boost your innovation capabilities and to obtain great results. So what is the National Competence Center? We offer guidance and support. We are the contact point for high-performance computing and data analytics. For industry, no matter the size, could be startup, SMEs, industry, to researchers from private and public organizations and public administration that would like to explore how to use HPC in order to innovate and to integrate into maybe larger research and innovation projects to help their organizations to be more competitive. We are also part of a European network of competence centers in HPC across Europe. Thanks to this network, we are able to exchange best practices and also to reach out to other competence centers and other partners across Europe to understand what's the best use of resources, or what are the best codes, or how to improve better the codes or applications or softwares that are available out there. We can explore for you what options are out there. In fact, there are also other actors, such as other projects, such as the Centers of Excellencies. They are more domain and sector focused, and they are improving available and open source codes and helping and supporting HPC users in specific domains. But let's focus on what we have in Luxembourg. So who are the players and the actors behind the National Competence Center in HPC in Luxembourg? Well, there is Lux Innovation. We support companies and not only actually to innovate. We help them to explore funding opportunities and to find the right partners for their innovation projects. There is Lux Provide. They manage the National Supercomputer Meluxina. They are focusing on industries and SME to democratize and help out industries to make the best usage, to include the users of HPC in their activities, to be more competitive and prepared for the future. They offer not only the usage and access to HPC resources, so to Meluxina, but they offer also IT support in case you need and you are not so experienced with the usage of HPC. So if it's your first approach to the supercomputing world, they are there to support you, understand how to access, how to improve your codes, and how to optimize your usage of the resources. Then we have as a partner also the University of Luxembourg. They also have HPC infrastructure, and they offer education and training. They have domain experts, and they are available for helping you out if you would like to explore research domain of your project. They could partner with you for setting up a research project. Then, let's say that what we have in Luxembourg is not just the HPC resources. So the first step was putting in place the HPC infrastructure, but we would like to, and we are building an overall HPC ecosystem to ensure that these infrastructure, these resources are used efficiently and could benefit companies and researchers as well, and to make the best use possible of what is available. So with an idea of creating a holistic approach and support, we help with operational and hands-on help. So thanks to its provider or the university, they have experts that can help you and consulting with what it takes to make your project on HPC work. We help you explore if there are financial support, so of opportunities, either at the national level or at the European level. Then we offer support for searching partners if you want to set up broader research and innovation project, and we make you liaise, put you in contact with other actors, other initiatives at the national European level that could be relevant for you and make you be or reach the success of your project. So as I mentioned before, we offer business support. We help you identify the opportunities where you can apply HPC to your activities. In fact, it could be not so easy if you haven't already done something similar. So it could be helpful to have any guidance from experts or just to guide you through the possibilities. The innovation agency, Lux Innovation, is here also to help you funding with opportunities and partner search, and our teams help you set up your projects, research and innovation projects. We help you with this technical support. So if you want to try it out or explore more, we offer the possibility to set up feasibility studies or to try out with a small project and set up proof of concept. We ease your access to HPC resources, and as I mentioned before, whenever you need some help with optimizing your code or understanding how your code can be adapted better or to obtain better results, of course. Then we don't stop here. We would like our HPC users to become independent. So that's why we offer training courses in HPC. The University of Luxembourg has its annual HPC school with hands-on lab, but LuxProvide is also offering trainings and could also offer customer-specific trainings. If in your company, in your organization, you have a group of people that are interested to explore and work with HPC, they are able to offer you tailor-made workshops and trainings just for you. Then, thanks to our European outreach and being part of the European network of national competence centers, we are able to put you in contact with the right people and find you international partners whenever they are needed for your projects and make sense to you. So Lux Innovation is your single contact point. If you like to have a meeting to discuss, to explore the possibilities that could best apply to your business, to your research, and to have guidance and support from our side, we are open to it. So don't hesitate to contact us. I will leave my contact details so you can reach out to me. And I will be able to support you, to help you understand the HPC ecosystem at the national and European level, to put you in contact with the right person for answering the best your needs. Now it's time for our Q&A session. Let's see first the poll, and then we'll see the question. We have a new question in the poll. This is about how do you evaluate the level of information provided during today's session. If it was easy to understand, then you got useful information, or it was a bit challenging, but still you got the main points, or it is too easy for you, you already know everything, or too complicated and you struggled. I see that 83% found this session easy and with useful information, and 17% found it a bit challenging. Well, we'll make our best for the next session to bring down the level of complexity of our session so that everyone can follow and get the main points. Then I see that you hear about the webinar from different sources. So we'll continue reaching out through all these channels. And that's 45% of our audience actually is already using a supercomputer. That's why maybe you are following everything easily, let's say, and 56% not sure if they will use or not in the future. So that's the perfect place to be to understand if it makes sense to you. And the last question, for what would you use HPC? We have variety of usage, I would say, from simulation and modeling to artificial intelligence to virtual testing and prototyping and other applications. Nice. Now I would say let's go to our Q&A session. So let's take the first question. Are there similar studies to yours looking at the effect of antibiotic treatment on the microbiome just after a treatment and or later in life? Eleftheria?
Yes, there are studies that they look into the antibiotic treatment. I don't work on samples with antibiotic treatment, but I know that there are a lot of other groups that they work on the effect that the antibiotics have.
Great. I think, yes, in your studies, in your papers, you need to take into consideration also what's out there already.
There are actually a lot of studies that they look into it, because the problem with the antibiotic usage so early is that it actually contributes also to the antibiotic resistance, which is another huge subject of the microbiome. I don't work with the antibiotic resistance though.
Okay. Thank you. Then another question for you. Can you say something about how much the runtime of the sample analytics was reduced when running on HPC versus your laptop?
Massive.
Massive.
So basically, it's occasionally I also work on side projects with animal samples, which is low number. Let's say 20 samples, for example. So if I have to try and run 20 samples on a computer with full processors using, the computer will probably take also overnight, especially if I want to be able to use my computer, because when you are running such analysis, your computer is slowing down. So if you want to do other things on the laptop as well, it's not very good. But 20 samples on the HPC, 2 hours, everything is done. So it's a very big difference.
Yeah, and usually you don't have just 20 samples, right? Because
no, usually,
as you mentioned before, the more data you have, the better your results.
Exactly. It's just that sometimes you might have some controls and you want to see if your experiment went well, for example. So you need to analyze to see if your controls are not contaminated, for instance. So, yeah.
Great, thank you. Let's go to the next one. So, Eleftheria, how were you trained to use HPC? Is that among the basic skills of all immunologists?
No, it's not.
Good question, actually.
No, actually, when I was doing my master in immunology, I didn't even know what the HPC was. The HPC appeared after I generated my first sequencing results, and then I was trying to do the analysis on the computer, and I was failing on RAM, and I was wondering what is happening, why I cannot do my analysis. And then eventually a very nice person came and told me, you know, this is a lot of data, you need to try it and go bigger. And this is how it all started. And how I was trained, as you already explained in your presentation, Ramona, there are a lot of courses actually, and there is a lot of support in Luxembourg. So I took the HPC school, and I also had some support through the LIH staff that they had experience with it. There is also a lot of available free material in the internet. If somebody is interested, they can find. And I have to admit that it seems far more complicated, but when you actually do it, it's far more simpler.
It's great to hear. That's a good encouragement for our audience.
It's the truth. And the best part is that when you do the analysis on the HPC and then you submit your script and then this is doing it for you, you are free to do something else. So it's really efficient.
Yeah, that's true. Thank you for your answer.
Yeah.
We have another question actually. So did you get some budget from LIH or the university related to the use of HPC during your PhD, and how that worked?
No, I didn't get anything related to budgets. The possibility to work with the HPC, it's free and it's available, it's open for all students. So I didn't have to go through,
through any, to search for other funding opportunities, other funding resources. Okay,
exactly, yes.
That was for your project maybe, or for the funding of your research.
Yes, but the truth is, I mean, this is available through the university and through the LIH for the main reason that this is essential up to a certain point. So
yeah, that's true. Good, thank you. I don't know if there are any other questions. Yes, I see. There are. If free, is there some intellectual property shared with HPC? I don't know.
I don't know either. What are the contracts between the institutions? I'm just a PhD student who's doing the analysis.
I think here our attendee maybe is looking for some answers or questions, the security measures of the infrastructure.
Okay, well, this is another story. This is possible depending on. I mean, in my case, I can give you my example. The data that I showed you earlier, and I talked about the analysis, they're coming from a cohort, so we have ethics in it. That means that your initial ethics proposal needs to be optimized in a way that covers the possibility to work on the HPC. But I think that it's becoming a must in this time of
Sure. Thank you. So maybe what I could add about this GDPR or security privacy measures that could be constrained, or maybe some users or regulations could apply. Just for you to know that with Meluxina, as it is kind of different from all or almost all the other HPC centers in the world, because they apply secure and trusty environment, and they put this in place. And they also are part of a well-known international security certifications, such as ISO 27001 and 27002. And Also, they take the security, data privacy, and compliance to GDPR very seriously, as they are working with industrial partners and the industrial partners and also this health area that's very sensitive to IPRs, so intellectual property rights, or privacy and ethics. So they have put in place the secure environment for you to be able to run your works, your workloads without concerns about these features. So just I can give you an example. So if in classical centers and in public cloud, actually, you get access to machine per core. Instead, on Meluxina, reservations are made per node. So to make sure that there is a hardware security enforced on the machine. And not to mention also another feature that the data center that is shared with Meluxina is hosting one of the digital embassies in Europe. And of course, security and privacy are important to the embassy and embassy environment. And this security environment of Meluxina is up to the level to respond to these standards and to ensure all the security measures that are needed. I hope this helps, and some of our audience attendees to reassure them. But if you have any other doubts, don't hesitate to reach out, or if you have any other questions, don't be shy. Let's see the timing. Maybe I have another question for you. So you told us you took the HPC school. So usually the HPC school runs by around a week, right?
Yes, usually.
Was it difficult for you to have this jump from Excel to the HPC, and how was the learning curve? Was it steep? Was so difficult to code, the coding part, the coding, how it was?
So the HPC school, actually, it's usually the first days they are for more beginner friendly level, and then the later days they're a bit more advanced. And I have to admit that I took the school twice, because the first time I was very first time, okay, I need to understand how do I control my space, my node, how do I do my reservations, how do I launch my scripts, far more basic things. But there is a lot of support from the team, and I took it the first time, and then 6 months later I was thinking, okay, I think I'm ready now to actually look at the more advanced things. And this is why then I retook it the next year, because I wanted to look at the more advanced possibilities that you can do within the clusters.
Perfect. Thank you, Eleftheria. So keep in mind that there are also some softwares available out there. There are also codes already there that you could adapt to your specific job and your specific project. And there are also softwares available on the HPC, or if there are not already there, as on your laptop when you acquire it, there are some software, but you can also install other software. So depending on what you are doing with a computer, supercomputer, there is this flexibility, and of course, as you mentioned, Eleftheria, there are some people that could support you to
exactly. In my case, every time I needed to do something that it was difficult for me, I would look out if there was somebody who did it before, and then it just takes one person to explain to you. You have to do A, B, C, and D, and then you can do it yourself. It's something that you can repeat it far more easily.
Mm-hmm.
And also something I think that it's worth to highlight, because you said the example how it was to go from the Excel to the HPC. When you're working within the HPC settings, your results, they're always far more reproducible than when you're doing them in Excel. So this is something that's worth noting.
What do you mean by reproducible? That you could run?
So Excel, you have an actual user who's actually working with the tables, okay?
Mm-hmm.
But when you work on the HPC, you work with scripts.
Mm-hmm.
So as long as your directory is well defined, then if someone else wants to reproduce your results, they would run the script, they should get the same results. But if you took two different people to work on the same Excel sheet, it is likely that you will have differences.
Yeah, that's true. Thank you. So I guess I don't see any other questions in our questions tab and neither in the chat. So I think we can go towards the conclusion of our session today. Therefore, I invite you to register for the next session and don't miss them. You can also watch the replays actually of our previous sessions that we had on the Lux Innovations YouTube channel. If you're interested to know more about how you could use HPC in your domain, we have a repository of HPC demonstrators at the university. I will just copy the link and paste to the chat if you are interested. I can also share with you the link to our YouTube channel so that you can you can find more easily the replays to watch. This session also will be uploaded to the YouTube channel of Lux Innovation in a few hours from now, but you will find the post on our social media, so don't hesitate to follow EuroCC Luxembourg on LinkedIn and Twitter. And what else to say? Thank you to Eleftheria. It was an amazing session. I'm happy you accepted our invitation. It was inspiring, and it's also a good example of how you could approach the HPC world and take the best advantages, take the best that you could to boost your innovation capacity, your competitiveness for the future, and what else to say? See you on 7 April for the following webinar. Thank you again and see you very soon. Bye-bye.
Thank you. Bye.